In this movie, I'm going to describe the sensitization of the gill withdrawal reflex in the sea slug aplysia. This is a simple form of synaptic plasticity mediated by a metabotropic receptor. Eric Kandel and his colleagues have used aplysia as a model system to explore the neural mechanisms that underlie learning and memory. They chose this particular animal because it has a simple nervous system composed of a limited number of neurons, but the neurons are exceptionally large. Some are so large that they can be seen with the naked eye, and the same neuron can be found again and again in different animals. Kandel and his colleagues conducted hundreds of seminal experiments on aplysia and discovered many of the cellular and mechanisms that can account for learning and memory. For this achievement, Kandel was awarded the Nobel Prize in Medicine or Physiology in the year 2000. Here I will describe a simple form of synaptic plasticity called sensitization that Kandel discovered in some of his earliest studies many years ago. As we shall see, sensitization is caused by the actions of metabotropic receptors and is revealed in the gill withdrawal reflex. Before describing the reflex, I point out the major structures of aplysia that we will be concerned with. The first structure is the siphon, a small fleshy spout above the gill used to expel seawater and waste. The second structure is the gill, the respiratory organ. And the third is the tail. Aplysia has a reflex for withdrawing its gill when the siphon is touched. It will also withdraw its gill if the tail is stimulated with a single electric shock, but the degree of the withdrawal is much greater than occurred when the siphon was touched previously. If one knock comes back in a few minutes after shocking the tail, when the gill has recovered, and once again touches the siphon, the gill now withdraws much more strongly than it did when the siphon was touched before the tail was shocked. In other words, shocking the tail made the siphon much more sensitive, so that when it was touched again, the touch evoked a far larger response, and the enhancement lasted for minutes. So next, we're going to see a demonstration of sensitization of the gill withdrawal reflex, but now in a real aplysia.
Looking at an aplysia stretched out, we're going to focus in on the gill and the siphon. We're going to apply an extremely weak stimulus so you can see the amplification with sensitization. Weak stimulus to the siphon, you see a modest withdrawal of the gill. Now we're going to frighten the animal, startle it, give a noxious stimulus to the tail. That, of course, causes a contraction in its own right. This contraction lasts for seconds, but you can come back minutes later. The same siphon stimulus now produces a much more powerful withdrawal. We can now compare the two, normal and sensitized, and see how much more powerful the withdrawal is in the animal that is startled.
Kandel and his colleagues then worked out the circuitry of the withdrawal reflex. The players here, of course, are the siphon, the sensory neuron from the siphon that innervates the motor neuron. The motor neuron then innervates the gill musculature. Another player, of course, is the tail, and the sensory neuron from the tail, which branches to innervate both the motor neuron and another neuron shown in blue called the facilitating interneuron. The facilitating interneuron uses serotonin as a neurotransmitter and does not innervate the motor neuron, but rather innervates the axon terminal of the green sensory neuron from the siphon. It is that synapse the synapse made by the serotonergic facilitating interneuron and the axon terminal, which is the key player in the sensitization. We now consider how the circuit actually works to generate sensitization. The circuit that was just described is shown in the top right of the figure, and an expanded version of the key synapse made between the facilitating interneuron, the axon terminal of the siphon sensory nerve, and the motor neuron is also shown in the center of the figure. The major players here are the serotonergic facilitating interneuron, the serotonin receptor in the axon terminal of the sensory neuron from the siphon, adenylate cyclase, voltage-gated calcium channels, protein kinase A, abbreviated pKa, voltage-gated potassium channels in the axon terminal, and last but most important are a second type of voltage-gated potassium channels called S-type potassium channels because they are closed by serotonin, hence the name S-type. It is this region of the synapse that the events occur that generate sensitization. Now imagine that the siphon is touched, which generates an action potential that invades the axon terminal. I would just like to emphasize that the repolarization of the action potential is due to the opening of all the voltage-gated potassium channels. Remember, that is what potassium channels do. They get rid of positive charges inside of the cell and thereby repolarize the membrane. The repolarization is shown here in purple. But before the repolarization occurs, the depolarization caused by the invasion of the action potential opens voltage-gated calcium channels, which then causes transmitter to be released from the axon terminal. The transmitter opens ligand-gated channels in the postsynaptic cell, the motor neuron, thereby evoking an excitatory postsynaptic potential. Now the tail is shocked, and an action potential invades the facilitating interneuron, which causes the release of serotonin from the axon terminal. The serotonin binds to the serotonergic metabotropic receptors. They are the metabotropic receptors. The binding of serotonin initiates a cascade that activates adenylate cyclase and ultimately phosphorylates the S-type potassium channels, causing the channels to close and remain closed even when the terminal depolarizes. Finally, the gill recovers and the siphon is touched again, generating another action potential that invades the axon terminal. But now the action potential has a much longer duration than it had previously. The reason is that the S-type of potassium channels are closed. There are now fewer potassium channels available to contribute to repolarization. Consequently, the action potential is prolonged because it takes longer to repolarize the membrane. The prolonged period of depolarization also keeps the voltage-gated calcium channels open for a prolonged period, thereby enhancing the amount of calcium influx. Remember, it is the calcium that triggers the release of transmitter, and since there is a greater calcium influx, there is more transmitter released, which in turn generates a larger postsynaptic response in the motor neuron and hence a stronger gill withdrawal. In short, the shock to the tail caused serotonin to be released from the axon terminal of the facilitating neuron, which then triggered a cascade that closed S-type potassium channels. The sensory neuron from the siphon was now more sensitive, in that the next action potential evoked by touching the siphon was prolonged, causing a greater release of transmitter and a stronger gill withdrawal. When these findings were first reported in the early 1980s, we were all surprised. We always thought that action potentials and axons were stereotyped responses, as described by Hodgkin and Huxley. Who would have guessed that the synaptic plasticity was due to a longer action potential caused by the closing of some potassium channels in the axon terminal? It is really quite remarkable.